Yes. Can I take this opportunity to welcome everybody to this public lecture on the theme of understanding brain aging and dementia, a life course approach. It's particularly good to welcome everybody here in Inverness and Ness Walk, but also to welcome everybody on who's out on the video links there. We have folk from a number of locations around the Highlands and Islands here for the lecture this evening. My name's Neil Simcoe. I'm Dean of Arts, Humanities and Business at the University of the Highlands and Islands, and it's a real pleasure and privilege to chair the lecture tonight and to chair the question session after the talk. But I particularly wanted to welcome Professor Lawrence Worley, who is honorary research professor at the University of the Highlands and Islands, Crombie Ross Professor Emeritus of Mental Health at the University of Aberdeen, and Professor Worley will be, of course, our keynote speaker today. Professor Worley retired in 2008 from the University of Aberdeen, where he was Professor and Head of Mental Health from 1992. Graduated in medicine and worked as a junior hospital doctor before training in psychiatry in Oxford and Edinburgh, and graduating MD Newcastle in 1969. Completed higher training in clinical psychiatry before being appointed in 1977 to the MRC senior scientific staff. And it was at this point that his work on Alzheimer's disease began. In 1986, he was appointed to the academic staff at the University of Edinburgh and established a research team to study brain aging in old people, with a special interest in the effects of high blood pressure. His landmark studies on the small area distribution of early onset dementia in Scotland prompted his current interest in the environmental contributions to dementia. After his appointment to the University of Aberdeen, he continued to work intensely on Alzheimer's disease. He's a fellow of the Royal Colleges of Psychiatry and Physicians in Edinburgh, was a member of the Scientific Advisory Board of Research into Aging, 2002 to 7, and continues to advise other grant-giving bodies, including the Wellcome Trust and MRC, on applications for scientific support. He's the author of more than 240 peer-reviewed papers and 39 book chapters. His 2002 book, a popular scientific account of the aging brain, was well received in UK and the USA before its translation into Japanese, Italian and Spanish. With John Breitner, he co-authored Dementia, Fast Facts, now in second edition. His current research programme relies on a population-based sample of 500 local people, all born in 1936, and who were IQ tested in 1947. This programme continues in Aberdeen in collaboration with Dr. Staff and Professor Alison Murray, and with Professors Deary and Starr in Edinburgh, and aims to determine sources of individual variation in brain ageing and progression to dementia. With BBSRC support, he has worked with the Rowett Research Institute in Aberdeen on the contribution of the nutritional environment to individual differences in cognitive ageing. From 2001 to 6, he held a Wellcome Trust Senior Clinical Research Fellowship. And since retiring, I think retiring is probably in inverted commas, from the University of Aberdeen, he's authored or co-authored 44 peer-reviewed scientific reports. He's currently contracted by the University of the Highlands and Islands to advise the principal on mental health research, and he has a new book with Columbia University Press entitled The Aging Brain and Dementia, a Life Course Approach, which will be published in 2013. So it's an enormous pleasure and privilege to ask Professor Worley to come now to offer his talk to us this evening. Thank you very much.
Thank you very much. second. So find this piece of equipment. There's a button. Thank you for your kind words. Could you switch it on for me? Well, thank you. I hope I can be heard in the distant parts. Thank you for your kind words. It's a pleasure to be here and a pleasure to tell you a bit about work that we've done in dementia and to explain the background to the Perth College research initiative looking at caring in dementia. The life course approach has been something that I've wanted to follow as carefully as I can for about 30 years, and it reflects largely on the way I was trained as a psychiatrist, to look at people in a particular way from early infancy to old age, to interview adults in the room but know that unseen to us all were their parents and grandparents and forebears influencing their thoughts and feelings at that moment. So the life course approach is really quite fundamental in the practice of clinical psychiatry. So it won't surprise people who trained with me that at this point in my career I'm returning again to the roots of clinical understanding to try and understand better how individual differences in brain aging and susceptibility to dementia might arise. This lecture is divided into phases. I have to apologise really from the outset that it's quite a complex subject. The brain is complex and we have about 45-50 minutes just now. And you know that if the brain were any more simple so that you could understand it, you'd be so simple you couldn't understand it. It is. It's quite difficult. So I've tried to seize upon some highlights and to move from one to the other, explaining as I go along. And there's about 15 minutes towards the end when I can take some questions and try and answer. First, I'm concerned with the idea that dementia may well be a developmental disorder, and it's not simply the effects of aging. I'll try and explain why. You need to know one or two basic facts about the aging brain, what's true, what's false, and when exposures that might have adverse effects on the brain arise, whether these happen by chance, just randomly, or whether they could somehow be linked, and so be like not a double whammy or a triple whammy, but multiple effects that accumulate across a lifetime. You're familiar with the term dementia, and many of you are familiar with the term Alzheimer's disease. What we need to know before we can study these things are they single ideas, are they single concepts, can they be defined with boundaries, such as we might define an object, or is it something more complex than that? You'll be interested to know whether or not, as we grow and develop as people, whether or not aging is a continuation of that development, or is it a phase of irreversible decline. And I've got to touch upon that at some point, because it's central to the thesis. You're familiar too that there's a great deal of publicity given to genetics and dementia, and that it's all in our genes, you might be told. We need to know just a little bit about that. And then what we need to know towards the end of the life course, is how people respond to the presence of dementia in their own brain or in the brain of someone they love. Living with someone living with dementia places particular demands upon us, and for many these demands are seen as a burden. And I want to think about that for a little while, towards the end, and ask whether or not everybody sees caring as a burden, or whether there are positive aspects of caring that we should pick upon and say, in so doing, this somehow makes us a better person. That someone who's lived a good life is deserved of a good death. That the person who's there to ensure that this happens is also gaining something from that. I want to touch upon that towards the end. Now, my acknowledgements, usually given at the end of a lecture, but I'm doing this at the beginning because I don't want to forget. These are some school kids in Aberdeen in 1932. They're in Skene Street Primary School. It's the nearest primary school to my office, so I picked this picture. And they're a cheerful bunch. I know about half a dozen of these children quite well, but I didn't meet them until they were 78 years old. And I did so because aged 11 in 1932, they were all born in 1921, they'd sat an IQ test. And I was using the IQ test as a baseline from which to judge any subsequent mental change across their lifetime. And then again, I've by now getting a reputation for collecting old people. This is a group at school in 1947, all born in 1936, who in 1947 set exactly the same IQ test. So by now, I've got access to records for IQ and recruited from the Aberdeen population over 780 of these children. And they became my life's work for the last 16 years of my time in Aberdeen, and the programme that was subsequently funded by the Wellcome Trust and others. What I like about this particular picture is there isn't an obese child amongst them. They've just come out of wartime. They've lived on rations. Some of them have endured the bombing and the loss of a parent in wartime, but they're a remarkable, resilient group, and may not be typical of old people that we encounter before or since. There may be something about having a lean, mean time. I remember asking one little girl, well, not little girl, she was a woman in her 80s, about going to school in the mornings in Aberdeen, and she walked a mile and a half. I said, that's 3 miles a day, that's 15 miles a week. Straight away, I went home for lunch. Some idea that these children were quite tough. Now the brain. This is a picture you'll be familiar with. It's a sideways-on view of the brain, divided into areas. It looks rather wrinkled like a walnut, doesn't it? This is the brain as it ages. It shrinks, and those wrinkles are separated more widely by little gullies. It looks like a wrinkled teacloth, if you can imagine that. And it's compacted into this box, which is our skull. If the brain, in fact, was smooth, we'd require a head 3 times the size. That would make difficulty walking, would topple over. But much more importantly, it would make childbirth rather. So we have this larger brain. By compressing the brain and folding it up, it means lots of things happen in the brain at a local level. This means more efficiency. I remember as a student, one of my fellow students became addicted to crumpets, which we thought was just an eccentricity, and was utterly dismayed when his supplier, Woolworths, stopped supplying them in April. And so he began asking for crumpets in Woolworths, insisting they got some more. They didn't respond, and he went to the corner shop, and by Friday the corner shop had crumpets in abundance. As we were leaving at the end of term, in the June time, Woolworths started stocking crumpets again. Now, what this means is that local services, in fact, can respond really quite quickly, and that's how the brain works. It works so efficiently because it can concentrate in localized networks a great deal of processing power and then share it as need be at distances. It also means that communication within little local areas of the brain can be very fast indeed, and that makes our brains so powerful as computing devices. One of the interesting things about the brain as it matures is that the wiring of the brain becomes faster by becoming insulated. It's a process called myelination. Very fast, very quick. The process of myelination occurs in the first 20 years of life. A strange thing happens in dementia. In Alzheimer's disease, this process goes into reverse. The first parts of the brain to be affected by Alzheimer's disease are the last parts to mature, aged about 20. Isn't that interesting? We almost go into sort of reverse developmental phase. When we begin to look at the development of the brain, we can find comparisons between ourselves and other species. You know, the Human Genome Project has identified how genetically different we are from chimpanzees. We're not really that different at all. We appear to have 98.5%, possibly more, genes in common with the chimpanzee. But none of you look like chimpanzees. You're not covered in hair. You're much, much smarter. Our brains are much bigger, 4 times bigger than a chimpanzee brain when you adjust for body size and weight. And imagine the moment that somehow the human brain has undergone a period in the last 500,000 years or so of accelerated evolution. And it's in those genes that have accelerated so quickly that people are now hunting like mad for genes that might cause dementia. And the reason is to do with this. It's called comparative neurology. And that is that amongst humans it's possible to make a diagnosis of Alzheimer's disease, of dementia, memory loss, selective areas of the brain lose brain cells, and abnormal proteins clump up inside the brain. When we look at animals, we can see that animals grow old. We've all had pets that have grown 12, 15, 20 years old. They become senescent. They're more frail and less able to organize themselves. But their brains never show selective loss. They never have those abnormal clumps. In fact, in all of the history of neurology, there's not been one single example of an animal developing Alzheimer's disease. There are some rare examples of the clumps appearing in some animals, but never happens. But of course, we live peculiarly long lives. The longest a chimpanzee has ever lived in captivity is age 67, which is actually only on the cusp of the age of highest risk for developing dementia. So there may be another explanation that we haven't worked out yet why this should happen. The other matter is that it's always assumed that we need to hold on to our grey matter in order to keep our faculties intact. Yet we can see here in this slide, across the first 20 years of brain development, we lose grey matter. We see that on this slide, which shows brains adjusted for body size, so they all look the same size brain. As it becomes more blue, so it's losing grey matter volume. And that's to do with the fact that it's the wiring of the brain that gives us a great deal of our computing power. The white matter, the big nerve tracts that communicate between one area of the brain and the other. It also means, too, that when we disrupt those white matter tracts with little bleeds, micro bleeds into them, so we disrupt communication. The brain, with aging, becomes disconnected. And it's that disconnection, leaving parts to function in isolation, that underlies much of what we call dementia. So what things can we say that are true or false about dementia, about aging, the aging brain? Well, the things that are true is that some degree of brain shrinkage is common, but that degree is always slight in the absence of dementia. That in normal, healthy aging, the brain shrinks a little. It's very, very difficult to demonstrate that brain cells are lost. The average brain cell shrinks a little in size, about 10%, meaning the overall size of the brain shrinks. But we're not losing brain cells in anything like the numbers that were imagined 50 years ago. And the reason is quite simple. It's to do with the way brain sections are prepared. And when we look at a microscope section, we see the brain cells that are closest to us. We don't see the next layer and the next layer down. The modern methods, in fact, can get a three-dimensional picture and show many more brain cells survive. The other thing about mental decline is there is a slight degree of mental decline up to about age 67, maybe later, as I'll come on to in a moment. It doesn't really quicken until we're aged about 67. That's true, but it's only a slight degree of decline. What's false is that brain cell loss always happens with age. The other idea is that all brain cell loss, when it occurs, is caused by hardening of the arteries of the brain. That also is not true. The large arteries are not responsible for what happens in most brains. It's tiny microbleeds in the tiny capillary bed that is to blame. Alzheimer's disease is not the same as brain aging. That's rather a brave thing for me to say, but I believe it to be true. There are some people who believe that Alzheimer's disease and brain aging are on the same spectrum, but the evidence is that it's actually quite different from brain aging. There are some people, though, who do very large population studies and say it's just shades of grey. Alzheimer's disease is just one end of the spectrum. The other idea is that it all goes together when it goes. That is, when you start to lose it, you start to lose it in every aspect of mental life. And that's not true. It's one or two areas that go. Eventually, many more will go, but it doesn't all go together in step. So that there are slight change is sufficient for expert clinicians to distinguish between different types of dementia. In fact, those differences may be more marked than a pathologist looking at the brain would accept occurred in life. So there's something about the way the brain functions, something about what the pathologist sees, they don't exactly correlate. And that may be because what the pathologist sees is an end state. That is after death. But what we see when we see living patients with early dementia is somebody living with the presence of dementia in their brain 10 years before they die. And what's left when they die may well be, well, the ghosts of things past, the ghosts of dead cells. But more importantly, it may be that because you lose so much tissue in dementia, you don't actually know what's actually happened. I sometimes liken the brain the pathologist sees to a visit to a scrapyard, where you see lots of broken down, damaged, rusted vehicles, and you're wandering around, and somebody asks you, what was the cause of that accident? Or this accident? The accident occurred 30, 40 years ago. This is the roadside wreck that's been brought in. It's very, very difficult, with hindsight, to look back and determine the very early changes that led those brain cells to die. And that's part of the problem in Alzheimer's research. Until recently, most of what we could learn about brain cells becoming diseased with dementia could only be learned by looking at the dead cells. And they were the survivors. You never saw the long dead cells. They were since gone. This is the life course approach. And I'll come back to this slide in various forms over the next few minutes. See, from age 0, on the bottom of the horizontal axis, when people are born, the brain development, cognitive abilities, that's the vertical axis, increase gradually. You'll see something called critical periods there on the left. And these are genetically determined, they're evolved very carefully over evolution, and they allow particular behaviours to be acquired during particular time points of infant development. Every mother knows about these things and knows, playing with a child age 3 or 4, how to do a jigsaw with that child. Say, oh, we look for corner pieces first. There's a bit of blue sky, that'll go over there. And she knows when to step back a little bit and let the child pick up the pieces, because fine motor control is developing. And the child can now match the picture to what they want to do. They can plan ahead. And the mother knows that. And in critical periods, for humans particularly, but in all animals, they all need to learn from their parents in some way or another. And their brains are prepared to do that during particular times. So when we learn to read and write, we do so to our advantage before age 10. The primary school teacher knows how difficult it is to catch up again if you miss an early opportunity to teach. Critical periods are under genetic control, and one of the exciting things to come out of the study of the biology of critical periods is that you can switch them on and off using genes. Does that mean you can, in fact, get people to relearn things later in life? And it's actually being discussed at the moment how you could actually wipe the slate clean and start all over again. Over the life course, here you see an adult life, some yellow vertical arrows pointing upwards and some multicoloured arrows pointing downwards. The vertical ones are things that hang together. I'll explain this in the next slide. The ones going down are independent things, but they accumulate over life, and they knock you back a bit. And here you see the broken red line showing how exposure to adverse things can in fact impair not only brain development, because it also deplete the resources available to brain to do its work. But they're removed, and natural aging supervenes, and the broken red line continues. And eventually, it crosses a horizontal blue line, broken line, which is the first symptoms for dementia, and onwards towards death. And what we see here is the effect of early life exposures having an immediate effect on brain, there being a long life period, eventually aging takes its toll, and one crosses symptom threshold earlier than one would have done had one lived to be 150 years old. You get the picture. If you live long enough in this model, you'll develop dementia. If you don't, you'll in fact develop it much earlier. This takes us to caring and end-of-life issues, which comes at the end of this lecture. Now, this is what I meant by linked exposures. I'm sorry it's a busy slide, but that's the way things are. The top part of it shows the life course from living in the womb into uterine, infancy, adulthood, and late life. Then we look down the left-hand column, you see poor antenatal care in the womb, impaired development of the child, impaired brain development. And then in infancy, poverty, poor diet, non-accidental injury, poor parenting with low stimulation, less education, impairing brain development. And there, a broken box called conflict and loss. And that box could operate right the way across the lifespan, but I've just put it there for clarity at the moment. Then in adulthood, we see some things that do cause the brain problems. Too much alcohol, smoking, obesity and abnormal control of energy metabolism. And part of that is to do with the failure to acquire healthy behaviours. That is, you can start smoking, but if you're smart, you stop. So when you look at smokers, the ones who continue smoking through adulthood are the ones of lower original intelligence to begin with. Most people recognise early on that smoking is not a clever thing to do, it's also very expensive, so give it up. We also know that the acquisition of healthy behaviours is closely linked to the acquisition of coping strategies. That people who adopt a healthy lifestyle have better coping strategies with adversity, they're more resilient. And they also find fun in cognitively demanding, mentally effortful things, which might make people with lower ability rather anxious and tense. But this won't surprise you, because you come along in the evening to a lecture. You're not at all surprised that there are people who get fun about having their brains stretched just a little bit. And there are people like that. Now, those people appear to be less susceptible to dementia. So if you forget everything else from this evening, remember, the sheer effort of coming along and trying to understand this chap at the front of the room, in fact, was doing you some good. But in fact, it reflects a lifelong habit of learning that you've taken into adulthood. Then in late life, we can see badly controlled hypertension, untreated hypertension, poor diet, particularly low dietary folate, predisposing to brain blood vessel disease, small vessel diseases of the brain, cerebrovascular disease. It appears in a way we don't properly understand to trigger some types of Alzheimer pathology. I'll come on to this in a moment. We also know that those things going on in childhood affect the capacity of the individual to buffer, to resist, to compensate for the presence of dementia in brain. I'll explain that in a moment. That is that as you develop a brain, so the brain, in fact, can handle a lot of what's thrown at it, including dementia. But first, we've got to get our minds around the problem of what is dementia, and whether it's a single thing or a composite. On the top part of this slide, I've divided dementias into three types. The central green one is Alzheimer-type dementia. The one on the right is the red one. That's dementia caused by diseases of the brain blood vessels. And on the left, a blue one, dementia attributed to Lewy bodies. Lewy bodies are found characteristically in Parkinson's disease. They appear to be different from Alzheimer's and blood vessel disease. They're the three major types of pathology in the aging brain. In the next row down, you see I've allowed them to overlap. So the dementia with Lewy bodies overlaps with Alzheimer's. So in the overlap area, there are people with both. And then brain blood vessels overlapping with Alzheimer's, and the overlapping people with both Alzheimer and brain blood vessels. And then in the bottom one, I've shown major overlap between blood vessel and Alzheimer-type dementias, and a minor, lesser overlap with Lewy body dementias. The bottom one is true. That is what we see in surveys of brains of people dying in late life, whether they have dementia or not. There's a blurring of these things. Pure Alzheimer's disease is relatively rare. Most common is a mixed type with both vascular and Alzheimer changes. And some people even have all three, Alzheimer, Lewy body. So this is not quite what the people raising money and shaking hands at the end of the supermarket checkout saying money for Alzheimer's disease, in fact, we'll be talking about. They want money for their target disease. The reality is much more confusing and blurred than that. Looks a bit like this. On the top slide, you see a green crescent on the right, a red crescent on the left, and a mixed blurred crescent. And that's roughly what we're seeing. And Alzheimer's disease is generally referred to as that blurred picture in the middle. They're all mixed up. There are midlife and early life influences on that, and there are effects on the right called cellular aging. We don't properly understand how these three things are linked together. Research is needed. This isn't just a concoction I produced the other night in an armchair with a glass of whiskey by my side. This, in fact, is adapted from the British Medical Journal just a couple of years ago from Marcus Richards and Carol Brain, who thought long and hard about this, saying we don't really know what we mean by Alzheimer's disease. Now let's move on to trajectories. This is a smooth trajectory of crystallized intelligence. This is why when you're age 60, they pay you all that money to run a business. Because you, in fact, you know where all the bodies are buried. You know lots of things. You may not be as fast on your feet, but you know so many different solutions that work. You don't need to think about a new one each time. You just go to the archive and pull out the one that works. So fluid intelligence, speed of thought, abstract thinking, tends to decline a little bit as we grow older. I mentioned reserve, and reserve is shown here as someone with high reserve and someone with low reserve. And I've shown a flat line of cognitive development, and that as dementia increases across the right side here, someone with high reserve will develop the symptoms of dementia much later than someone with low reserve, but they'll both arrive at the dementia endpoint at the same time. There appears to be a lag period, a delaying of dementia onset with people with high reserve. So what is reserve? What provides it? Well, the structure of the brain appears to be important, so people like Jakob Stern will say there's lots of compensatory mechanisms there. Imagine you had a computer at home and you opened it up and exposed the motherboard, and you poured 25 mils of water onto it because it was alive. It would go kaput and the computer would cease to work. You could come back 6 months later and turn it on and it still wouldn't work. If you take a human brain and you allow a bleed into part of it in a stroke and you watch what happens, the brain doesn't work so well. A dense paralysis may follow with loss of speech, and it looks as though it's generally kaput. Come back 6 months later, and the person is speaking normally, recovered a lot of their motion, mobility, and voluntary movement. They're not the same. And it's as though they've recovered, and the nursing staff have been helpful and positive, and the physio and the speech therapist, and they've rebuilt this person around. But you take an image of the brain, the still a big chunk, an octocube size of brain tissue is gone with the stroke, but they found a way around it. And that's something that a computer can't do. It can't mend itself. It can't find alternative ways of doing the same job. Brain reserve is rather like that, but it's more than that, because we find that brain reserve, cognitive reserve, has other determinants in early life. One is childhood intelligence. Smarter children appear to have more reserve, develop dementia later and less often. The well-educated, the highly educated, do better. People say to me, oh, what about Iris Murdoch? She was highly educated, very smart. So she was. But she may well, hadn't she not been so smart, have developed her dementia much earlier. It was postponed by the reserve. Education support. The complexity of the jobs that we follow habitually are important. So people who do complex jobs, often involving other people, often involving data, in fact, seem to do better cognitively in later life, getting less dementia. That's encouraging. Then there are two oddities that come out of recent research. The bilingual people from infancy, able to speak Two, mutually unintelligible languages have much lower rates of cognitive aging and probably of dementia as well. That's fascinating. And then there are the people with high musical ability and training. They also develop dementia a bit later. Now this has to be explained, and it's probably through acquired strategies in early education, no matter how people are educated and what they're educated in. Maybe even learning the Quran is enough. But it's the task of taking this on and experiencing mental effort. So it's no longer anxiety provoking, but in fact it's a source of joy and pleasure. This is some data that we collected, and I'm showing this to you because those smooth curves I showed you before for cognitive development aren't true. This is what you find when you go and find 750 people and you see them every year or two and do mental tests. It looks like this. You can do the mathematics on them, and you can show that there's a general decline, but it's slight. That's the solid red line. But there are people there who, as you test them again, they get better and better. It's not all a story of decline. There are individual differences. It's not the way you think it is. Just because you see a line showing decline doesn't mean to say either it's inevitable or it happens generally. There are exceptions. In fact, in our Aberdeen studies, we had some, mostly men, who came back from age 77 to age 90 and did better each time. They're not supposed to do that. One of them got himself into the Sunday Times. The reporter, a very young attractive lady, went round to see him at home, wanting to see who this modern-day Einstein was. And he was a retired RAF technician. And she said going into his apartment was like getting onto the flight deck of USS Enterprise. He had computers all over the place. He was in shortwave radio communication with people in the Pacific. And he was doing engineering tasks as he, in his late 80s, and getting great pleasure from them. He also had 4 girlfriends and 1 car. It's another story. How he got to this. He told me, I said, how do you manage? And he said, well, they're all called Margaret. So the explanation was that he just said, Maggie. And when Jack McArdle reanalyzed the data, he showed that up to age 67, 75, 80 is generally pretty flat. The decline really doesn't kick in until 80. Now, this point of this flexure in the graph has shifted towards 80 as Jack has got older. When Jack first came into his 40s, and the flexure appeared when he got 65, was as I've known him and recalculated. More data, he says, it's more data. But I think it's just his perception of when it's all going to happen. Genetics of dementia, timing of positive and negative effects. Again, a complicated slide. But if you can pick up here, there's age running across the bottom, cognitive abilities are vertical, and then there are all sorts of genetic things that contribute to things going wrong: schizophrenia, intractable depression, chronic respiratory failure, diabetes, and we could add multiple sclerosis, we could add epilepsy, we could add other things that predispose to lifelong disability and handicap, learning disabilities. How do they prepare you for late life? Are you more vulnerable because of them? If you're more vulnerable and they have genetic determinants and they're overrepresented amongst the, quote, dementia, then when you look for the genes for dementia, are you looking for genes for dementia or the genes for the vulnerability to the dementia, which may be quite a mixed bag of things anyway? It's a very difficult task. How we focused on it is to look at the pathology of Alzheimer's disease these last 30 years, looking for the proteins and asking by reverse genetics, what are the genes coding for those proteins? And they go hunting for them. One of the groups with learning disability that's predisposed to Alzheimer's disease are babies, children born with Down syndrome. Over 95% will develop Alzheimer's disease from age 40 to 50. That's terrible. But when a gene was found for Alzheimer's disease, the first place they looked was on chromosome 21, because it's chromosome 21 that is trisomy in Down syndrome, and there it was sitting there. But that is not an explanation that was later shown for Alzheimer's disease, but it was important in a small number. The genes for Alzheimer's disease account for about 30% of the dementia, the Alzheimer dementias arising before age 65. Those same genes account for 0.03% of dementias occurring after 65. It's a tiny, tiny number. So this is not the same thing. But it hasn't stopped people pursuing the genetic story, looking for the genes, looking for the pathways towards dementia. And one of the groups that's been doing this most successfully is in Colombia, in the Medellin province, in the Western Highland of Colombia, led by Francisco Lapel. And he's found a number of families, I think it's about 25, and he's extensively investigated their pedigrees because they have a form of early onset Alzheimer's disease. Those 25 or 30 families' pedigrees can be traced back to one founder, one immigrant to Colombia in the 18th century. They've now got over 5,000 descendants, of whom about 2,500 carry the same genetic mutation for Alzheimer's disease. And Eric Riemann in Phoenix, Arizona, has been flying these people up to Phoenix from Colombia and doing brain scans to look at the people at risk of Alzheimer's disease to see what is happening in their brains before they develop symptoms. And the reason for that is that people are now convinced that if we are to prevent Alzheimer's disease, prevent dementia, we've got to be looking at people at the very earliest stages, not when they first get symptoms, but 10, 20 years beforehand. So that the main task currently is to find ways of identifying those at greatest risk. But if you find them and they carry the gene, but you depend upon the disease starting to find them, are you actually preventing? No, you're coming in still too late. So people are trying to look for what happens beforehand. So the great prevention studies are looking for people in almost in late childhood, early adulthood, for the earliest signs. This is a diagram I'll show twice, once before I finish. This is from New England Journal of Medicine in August of this year. And this group, the Autosomal Dominant Alzheimer's Investigation Network, have been looking at families with family members who carry genetic mutations that predispose them to Alzheimer's disease. And they've looked at family members who carry the mutation and those that don't. And they've measured things that might predict Alzheimer's disease. And these are the things that they've traced. And they've got a time, which is the vertical broken line in the middle, when they're expected to develop symptoms. And for up to 30 years beforehand, they've got values here for things that they can measure in the blood. They can actually image the abnormal protein deposits in the brain using a type of PET scanner. And they can also measure the volume of key structures in the brain involved in memory. And they can chart these out. And what we see here are very gradual changes over 10, 20 years before expected time of onset, age of onset of Alzheimer's disease, beginning to change. So using people at high genetic risk who are known to be almost certain to develop Alzheimer's disease before age 70, they've shown biological changes. But of course, the task is saying, well, at what point in the normal population would these measures predict that we're going to get Alzheimer's disease? And of course, there's so much variability up to about 10 years before, you can't be certain at all, even by combining them. So this sort of research is in its infancy, but I tell you, August 2012, New England Journal of Medicine, this is as recent as it gets. This is as good as it gets in terms of prediction. I'll skip that slide, it's a bit too complicated. This is less complicated. the red banner in the middle looks at development again, which I talked about earlier. It starts with parental genes, it goes to low birth weight, it goes to lower childhood intelligence, it goes to those bad habits of adulthood, smoking, poor diet, social isolation, all predisposing to increased cognitive aging. It's down the middle. At the top, there are things going wrong in utero. Poor placenta. Something called epigenesis, which is the way the environment modifies the genes a child carries, whether they're expressed or not. And then other things happening in childhood: illnesses, education, occupational training. Adulthood, being exposed to solvents at work that they're thought to be important. Some types of toxin derived from plants is also thought to be important. Non-insulin dependent diabetes in later life. Obesity is important. This is the picture. It's the picture that we produced as a research group about 6 years ago, and it remains the map that we're currently using to try and understand what's happening in the life course, because what the map has are points where you could intervene. You could intervene in better antenatal care. You could intervene early on when the brain is developing in childhood during education. You could intervene in health and safety at work and occupational toxins. You could intervene looking at how to reduce obesity, poor exercise, social isolation, high risk of heart disease in midlife. All of these things can modify dementia risk. These are our opportunities, as we understand them currently, for modifying the most common forms of late onset dementia. So these are the fetal origins of diseases of aging: adversity in pregnancy, things happening in brain development, and a sum total of things to do with brain reserve that is there to buffer against the presence of dementia. And it's a building of brain reserve in our children and adolescents that's critical. We do not encourage experiments in adolescence like taking illicit drugs that kill brain cells. This is a bad idea because it probably diminishes cognitive reserve. It certainly lowers childhood intelligence, taking things like cannabis in your teenage years. Ecstasy, MDMA, and the like, are neurotoxins, and they take away chunks of brain tissue. We don't know the results of this experiment because the children who are doing it in their millions worldwide are not yet old, but we confidently expect that they're going to increase their dementia risk. This is going to keep people like me occupied for some time, but it's not a very clever thing to do. So one of the compelling reasons that to argue against the use of illicit drugs and the like is to do with the fact we don't know what the long-term consequences on brain are. We do know that when we do studies on the brains of children who take these drugs, we can demonstrate toxic effects directly on the brain and loss of brain tissue. Well, coming to the caring end-of-life issue in the last few minutes of the presentation, it's about the Perth College study. We're moving towards the end now of life, and we're looking at how people are cared for, and who are the experts in care. Well, the experts in care are carers. The experts in care are people who choose to work in residential care in hospitals. But when you dig into it, and you go into the residential areas, and you find out who are the culture carriers, who determine the ambience of the place, you find those are permanent members of staff, the ones who have been there some years. And the strange thing is they're almost entirely women. They're almost entirely women between the ages of 40 and 60. And they seem to be a dying breed because they aren't going into all these care numbers they used to. But they're the carers. But the other carers, the other expert carers, are families who've lived with dementia, sometimes for a generation or two. They know what to expect. And the Perth College study is about trying to learn from families who care for dementia. What are their special needs? What are their special skills? What are the problems they're faced with? How do they address and deal with the issue of their own personal vulnerability to dementia because they're part of the same family? So these things are intriguing us at the moment, and we're hoping that by studying and listening to their accounts of themselves, we will acquire new information that will be transferable not only to other families and other carers, but also to the educational milieu that's provided in Perth College for the next generation of people who move into the caring professions. So we want to know about families multiply affected by dementia. Are their needs different? Do they differ from families who aren't multiply affected? We want to look at how we can develop support programs which focus specifically on familial risk. on familial clustering. And we want to work with people in the genetic counselling services to try and structure in a better way how advice is given at a personal, individual level to families. We don't know how to do that yet. It's likely that the biological studies that we were talking about before will run ahead of us. This one from August 2012 is just one. More are coming. More studies will be identifying risk. It may well be that in advance of anybody working out what the needs of the families are, the biology will be telling people, we can determine your risk. What do you do with that information? Well, first and foremost, you've got to find out whether people need the information or not. And by giving the information, what additional problems you might cause. So you need a solid substructure of knowing about the families before you can apply the new genetic knowledge. When I talk to genetic counsellors about this, they throw up their hands in horror because there is so much dementia out there, and there's so much familial dementia. There are 5 centres in Scotland providing expert genetic counselling services. There are 7 consultants, and they're dealing with disorders mostly of childhood. They don't really want a queue of old fogies at the door asking about risk, because it has particular implications. So we want to, at this stage in the Perth College studies, somehow determine, if we can, the positive aspects of caring. We want to look beyond viewing caring as a burden. We know that not all families will feel the same way. We know from the literature that some families, particularly lower social class families with little education, cope better with caring than smart families. This is particularly true of the differences between African Americans and white Americans. African Americans get more out of caring. It sounds like a puzzle, doesn't it? And when you ask the black American, they talk about their responsibility. They talk about their religious beliefs. Is the same true in Scotland? We don't know. No one's ever asked. There are people who remain quiet at home as carers. We don't hear their voices very often. We hear mostly about people who perceive caring as a burden. We've got to somehow work with that. We need to learn from people who've learned themselves how to deal with things. It's what, in fact, are the important components of caring. Can we learn from families multiply affected by dementia? I'll finish on saying thanks to my Perth College colleagues, Alan Brown and Maggie Robertson, who's running the project in Tayside, and then my colleagues in Aberdeen, Alison Murray and Roger Staff, who you heard acknowledged earlier. Many thanks.
Thank you very much for what was a fascinating insight into the world of dementia, and I'm not from a health background. It was absolutely fascinating, and thank you very much for your input. We do now have some time for questions, either from folk from the video conference links, or indeed from folk here. So it's an open floor, and please press ahead with any questions that you would have.
Can I have a glass of water?
Any
questions? Please.
I just wondered about where does Pick's disease come into this? Is it a form of Alzheimer? Because I know that's more early onset of dementia. Is that a form of Alzheimer's as well? Or?
I'm being asked whether or not Pick's disease is a form of dementia, whether it's a form of Alzheimer's. Pick was a neurologist working about the same time as Alzheimer, and he identified what are called Pick bodies in the brain of some people dying with what was called a lobar dementia. Thank you very much. And that's the way it remained. It was regarded as a medical curiosity. In fact, in the early 70s, was one of the first disorders to be related to evolution. The most recently evolved parts of the brain were affected by Pick's disease. That was until the mid-80s when the Swedes began looking at these dementias, which were called Pick's disease as a group and distinct from Alzheimer's. So you say, well, they're earlier onset. And a group in Manchester, Tony Neary and David Mann, began to look more carefully and found that Pick's disease turned out to be a group of at least 4 or 5 different early dementias, amongst which the most important were the frontotemporal dementias. And then the genes for each of these were found. And then some of the genes were found in motor neuron disease. So this looked really complicated. So there was a spectrum of disorders with important genetic contributions called frontotemporal dementias, no longer Pick's disease, and accounting for about 10% of early onset dementias. We don't know the proportion of late onset dementias that are frontotemporal. We think it may be much less than 10%. When we look at the surveys, it's just a few percent. It's a much less common than Lewy body dementia. The interesting thing about these frontotemporal dementias is that they're relatively sparing of memory for the first few years, but they have, to me, fascinating symptoms to do with taking meaning from words, difficulty with language, and then a dementia supervenes later on. So it's been a very important group of disorders in terms of understanding the biology, and it's one that's been rediscovered in the last 20 years as being very important. But we're pretty certain that in terms of the common or garden late onset dementia that we usually refer to, it's pretty unimportant. But like all models for disease, it may be telling us something really quite important about how the brain adapts to age-related changes. Because underpinning all of this, like Huntington's disease as well, nobody can explain why the genes don't kick in until you're 40, 50 or 60 years old. Not, as you might expect, from childbirth.
Thank you.
What would be the benefit of early detection?
Well, without a treatment, it seems pointless, doesn't it? Mm-hmm. Unless, of course, you're the person who now knows that your life expectancy is seriously short. So the main advantage currently of early diagnosis is that it gives people a framework within which to organise their lives, to make what are called living wills, for example. This is how I wish to be cared for if I become demented. It also allows people to inform their relatives of the, particularly for an earlier dementia, that their genetic risk is increased also. And this means that some counselling can be extended family-wide. So there are 2 important benefits of early detection. We need to plan services as well. You're all, I presume, familiar with human variants, Creutzfeldt-Jakob disease from bovine spongiform encephalopathy. There's not one person in this room can tell me how many dementias are going to occur in the people in this room from their having eaten contaminated meat during the bovine epidemic of the early 90s. If I told you, half of you are going to get dementia by the age of 60, if you live long enough, you'd immediately take a bit of notice. So I said, well, I'd be surprised if there was one. You wouldn't be at all interested. So early detection is also very tightly linked to our planning of resource allocation in the future.
I've just got one question. Can I just clarify some of what you were saying in the slides, that if people are exposed to extreme stressful situations, not just for a short period of time, but over a number of decades, that this can actually start the process of Alzheimer's later in life?
This is called the Sapolsky hypothesis, and it derives first from primate research, but mostly done with rats. but it's also found in people who've had post-traumatic stress disorder and sometimes of severe mental illness. There are parts of the brain that are specifically needed for memory to function that are shrunken, they're damaged by stress. And the mechanism is poorly understood, but it's seen as being probably a major factor for lowering the threshold at which dementia arises during the aging process. So the idea is that acute stress, or as acute severe stress as in a battle situation, or chronic cumulative stress as in domestic abuse, that can make part of the brain, the hippocampus, smaller, and memory subsequently, as aging supervenes, is impaired.
Thank you.
Are there any societies in the world which have a notably low incidence of dementia, and what might you ascribe that to?
There are some that don't get dementia at all, Alzheimer's disease. The Japanese, for example, have much lower rates of Alzheimer's than they do of vascular dementia, and that's associated with their lean diet, high in fish oils. The sub-Saharan Africans are said to have lower rates, and when Africans in Nigeria and some parts of eastern Kenya are compared with black Americans living in, say, Indianapolis, they have lower rates of dementia. The same team have shown that Native Americans have lower rates of dementia in some provinces of Canada. So there is a geographically isolated tribe in northern Manitoba of many hundreds of old people with no recorded case ever of dementia. And this year, a number of communities in South America of native South Americans have been described without dementia. I do not know why.
So another question. In these communities, is no dementia and also no metabolic disease?
I can't understand the question.
In these communities, you mentioned there's no dementia. Is also no metabolic diseases?
It's very difficult because the isolated community living in a subsistence culture has evolved to cope with intermittent famine and hardship. And the genes needed to do that grab hold of every bit of energy they can, and they look after it. They are thrifty. They squirrel away bits. Now, if you travel in Polynesia and you go to some of the developed islands like Samoa, you'll be amazed at the very large size of the women and the even larger size of the men. And these Polynesians have migrated across the Pacific, and the survivors have been the founders of the populations. The weak fell by the wayside. Those who could cope with famine. But of course, when they arrive in New Zealand with a Western diet, they develop very large body sizes, and they have problems with diabetes, mostly, and renal disease, and gout. But that's fairly well known. But the vegetarian societies in India, southern India, they're much more interesting. They seem not to get to dementia as well, very low rates. But vegetarians in Europe have the same rates as we do.
Are there any further questions? Somebody on the video conference, I think. Yes, I think it's from Perth there.
Can you hear us?
Yes, we can hear you.
Professor, can I ask you to tell us more about how you contacted the pupils of the primary schools, and also will there ever be another opportunity to get IQ data?
The children of the primary school were children who were IQ tested by the Scottish Council for Research and Education in 1932 and 1947. And that was done because after the Great War, there was concern in Scotland that the cream of Scottish society, the menfolk, died on the fields of Flanders, so weakening the genetic stock of Scotland and reducing Scottish competitiveness for the 20th century. And to demonstrate this, they went out to look at IQ. The money for the study came from the Eugenics Association, and they planned to do it again in 32 and then in 36, but it was after the war before they could do it again. The test they used became the dreaded 11 plus when it was evolved. We were fortunate, I was fortunate, one Tuesday afternoon in March 1997, I found the records stored in cardboard boxes in the basement of a large building in St John Street, Edinburgh. And the council who ran that, their ethics committee, gave me permission to begin to contact these people. Of course, I couldn't do that directly. I had to go through the health board, who then wanted to check, and then eventually local general practitioners agreed to contact people. And I think we had an 80% positive response rate for volunteers the first time. And in the 1936 Perth College, we had 75% response rate. Aberdeen is known for people being motivated to do this sort of thing. So our study is not famous, but notorious for the fact that it contains a very wide spectrum of society, whereas many of the similar studies concentrate on the materially better provided for.
Fascinating.
And no, it won't be able to be done again, because no longer do primary schools measure the IQ of children. Is that true?
Yes. Although who knows what might happen in the future.
If Mr. Gove has his word.
Exactly, yes.
Given the wealth of data that you've provided to us this evening, I just wondered if you had a working hypothesis as to why, in terms of modern treatments like cholinesterase inhibitors, some patients responded particularly well and some didn't respond at all.
Well, the current working hypothesis is that what we call Alzheimer's disease, which is the diagnosis that's the first step to giving a cholinesterase inhibitor, in fact is not one disease but several. And that there is one disease where the primary disability is the manufacture and use of acetylcholine. But other types of dementia have other things present as well. Now, what this means is that the two things. One is that what we call dementia is an end state, a final common pathway, with many pathways leading into it, one of which is the cholinesterase route. But the picture looks the same in all of them, a bit like the scrapyard, looking at wrecked cars. The other idea is that it's likely that by themselves cholinesterase inhibitors are not enough. That what in fact is required for some patients is something in addition to that. And so the current thinking is that we need to combine something that addresses the brain cell dysfunction and the vascular problems simultaneously, since they clearly coexist importantly in the same patients. So that's the current thinking. And I suspect the people responding to cholinesterase inhibitors also differ genetically. There's another thing about acetylcholine, which I haven't talked about here, but I mentioned very briefly in the slide I didn't show. And that is that the cells in the brain that are most susceptible to death with aging are those that are most energetic, the pyramidal cells of the cortex. And it's likely that it's these cells that are being driven by acetylcholine. It's trophic in these cells. So that is an idea that you'll find in the literature.
Thank you. Just got an opportunity for a couple of final questions, if anybody would like to ask anything.
Could I come in, please?
Yes, please do.
I work in a health improvement role, and I came in partway through the presentation, so I didn't get the beginning, but I'm wondering what key messages I should take in terms of health improvement. I wonder if there might be a belief amongst the general population that there isn't anything you can do to prevent dementia and Alzheimer's, So I just wonder what the key messages for me are.
Well, in simple terms, which I think what health promotion tries to do is to get a message across that people can understand. The simplest slogan is, if it's good for your heart, it's good for your head. That is, if you do things you're told, like keep your blood benign, your blood pressure, keep your blood fats down, take exercise, don't smoke, have your blood tested for diabetes, urine tested for diabetes, those things are important. The other part of this is in health promotion, the acquisition of healthy behaviours in early life is critical. So what I talked about here were things, the habits that people acquire as children and youth. The need to take exercises, the need to make sure you're eating fresh foods that are nutrient rich but not energy dense. That's important to get across. And that's a major health promotion message. But it need not be specific for dementia. The main thing I think that's specific for dementia is probably to do with education. I suspect that, like all things, how you treat our children, how you bring them up, how you nurture them, lays the foundation of a healthy brain throughout their life. And that's where the trick is. So in health promotion terms, I think you go back to health promotion in the classroom. Back to primary care, where we were at the very beginning.
Fascinating. And my background is education, so we were just talking before about the overlap here. That's very interesting.
You didn't think there would be one, did you?
Thank you for your question.
Sorry, just one final one, based on slightly what you just said there. If a person didn't have a very good start in life under the age of 10, or even under the age of 15, but kind of educated themselves later on in life, in their early 20s, early 30s, is it still too late, possibly, by that time?
Well, the acquisition of healthy behaviours has its greatest impact on midlife behaviours. So you can throw quite a lot at your body in terms of unpleasant things early on. It's the lifelong habits between age 30 and 70 that are important. And they're the foundation of good health in later life. But I'm not saying anything that's dramatically sort of unusual in that sense. We all recognise that the chances of developing something serious are actually quite slight up to about the age of 65. And that most of the things that happen prematurely are avoidable. And if you use the health services well, you go and see your doctor and have a checkup, not asking for mortgage advice. You know, sorry, I'm not joking here. When free healthcare was offered to poor Americans, they in a trial, some didn't get it, some did. The ones that got free healthcare began asking the healthcare givers for financial advice, because they were the only responsible adults they ever met. So the doctors obviously know how to deal with money because they're so rich.
Thank you.
Thank you. Is there one last question?
In Perth?
Yes, please.
Professor, in view of the current concern with alcohol in our society, do you foresee maybe in 15, 20 years' time a significant increase in dementia?
Did you mention alcohol?
Yes.
Can I just check at the back? Did everyone hear the question?
No.
Do you want to just...
I'm being asked if the current wave of high drinking, where we've a lot of people are drinking much more than their parents did, is going to cause dementia problems. Well, there are two sides to using alcohol to excess. The first is you won't live long enough to get dementia. When we looked at our 1921 born people, age 77, we found very little alcohol used. And the main reason was, of course, those who used glass alcohol were dead, mostly from increasing risk of heart disease and cancer. So that sort of solves the problem in a way. The other is old people beginning to use more alcohol. And again, that's linked to premature mortality, from falls and the like. And mixing it with their regular medications, not a clever thing to do. So I don't foresee an epidemic of alcohol-related dementias. I see much more obvious medical complications leading to premature death from excess alcohol, from liver disease, from falls. It sounds a bit callous, I'm sorry, but I think that's a true picture.
Thank you very much. I think it's probably just about time to bring the event to a close. Can I first of all thank you, Professor Worley, on all our behalves for what has been a very interesting and thought-provoking session. So we do thank you for coming along to deliver your lecture and to engage in the questions. Thank you very much. Before we leave the various buildings, just to say that the next event in our lecture series is the University of the Highlands and Islands Christmas Lecture. I think this is the second Christmas lecture. This event takes place on the 6th of December at Eden Court Theatre here in Inverness. The guest of honour this year is Roy McGregor of Global Energy Group, and he will be interviewed on behalf of the university by writer and presenter Nicky Marr. There will also be some music provided by our BA Applied Music degree students. This is in my faculty. The first cohort of students coming through will provide some musical entertainment. As always, the event is free, and places can be booked through our events team. So we do hope to see as many of you as possible at that event. But thank you very much for coming this evening, and thank you once again for your lecture.
I think we've been switched off.